Hello, and welcome back. So, today we're going to go ahead and dive into chapters 24 and 26. I'm going to bounce around a bit between these two chapters. There's some things that I find that are kind of important for moving on with this, but keep in mind, this is also a lot of things that when they change in science, it's usually changing in here and our understanding of it, like, this is going to be the kickoff to going through the domains and then the kingdoms for the rest of the semester until we get to ecology. But for the most part, speciation of how species split off, we kind of touched on a little bit of some of the mechanisms behind it in the last lecture, but in this lecture now we're going to go on and look at some more examples further in, but also we're going to be going down the rabbit hole a little bit into how we do classification and how classification has changed over time and is still changing. It's one of the most contentious spots of science, especially biology, is usually the classification place, because if we finally just gave up and just classified things, again, it wouldn't give scientists that love to work on classification a job. They'd be out of a job, and that'd be sad, I guess, for them. For some of us, we kind of look at them and go, could you just make up your mind? But no. And there's a lot of different reasons for that. Most of the time, it is newer technology is coming in and giving us new looks. Plus, we're also finding, with newer technology, we're also finding new things because it lets us expand our horizons and get to places we couldn't look. But there's some areas like we still don't know exactly, where certain things came from. Sometimes we do. It's a changing landscape of how we classify living creatures on this planet. And it's one of the most variable things that changes almost year to year. And so I'm going to give you an overview, but if you ever want to take an evolutionary biology class in the future, highly recommend. Simply just to, then you think you know something and then you go on and the next year it's all changed, which has happened to me teaching several times. It's like what I was trained in when I was in college is not that anymore. They introduced some new things and that made me go, what? And I had to re-educate myself. And it happens a lot. That's the way of science. Science should be changing. If science doesn't change, we'd be in a whole lot of trouble. So remember, change in science is a good thing, even though sometimes you can turn your head for 1 minute and everything's changed and you go, oh. Anyway, so remember, let's review a bit and dig a bit deeper. So again, moving on, and of course it's now the cats decide to have their zoomies. So again, Darwin was fascinated by speciation. And this is a process by which one species splits into two species. And again, from what we talked about in the last lecture, there's two major things going on here. Microevolution, which consists of allele frequency in a population over time. So looking at genetics, whereas macroevolution is the broad patterns of evolutionary change above the species level. So microevolution looks at, the genetics on a species level, how that affects a population. Macroevolution is looking at the big picture with the population and the stressors coming from the outside. So remember, the word species is Latin for kind or appearance. And so comparisons of physiology, biochemistry, DNA sequencing generally confirm the morphology of distinct species in discrete groups. For the longest time, we only had physiology. And boy, did that lead us down some rabbit holes that did not work. And I'll actually probably show you a couple of what we thought dinosaurs looked like before we had, computer modeling and a whole bunch of physics, and we only had the bones, and we were going, how does this even go together? Again, I think I mentioned it in the last lecture. If you ever look up the Bone Wars, that also showcases where we got it wrong, because that gave two paleontologists a reason to fight over Bronchiosaurus / Apatiosaurus / where does the head go? It's actually kind of a funny story. I might get into that later. I wish I could show you videos without this being flagged on YouTube, but anyway. So, according to the biological species concept, a species is a group of population whose members, now this is important, have the potential to interbreed in nature. So, in other words, if we take one group of lions and another group of lions, they can make lions, and voila, we can still, even though they're living in different areas of wherever in Africa, they can still produce fertile offspring. And, and they do not produce viable fertile offspring when members of other groups. So again, an example of this would be like horses from different places. You can take a Clydesdale, you can take an Arabian. Sorry, I was raised in a horse farm. It shows. I was raised in a barn. When somebody asks me if I was raised in a barn, the answer is actually yes. If I take a Clydesdale and an Arabian and I crossbreed them, I will get somewhere in between a Clydesdale and an Arabian because they're both horses and I can do that. But if I take a donkey and a horse, I'm going to get a mule. And a mule is not fertile itself. To make more mules, you need more donkeys and you need more horses. Even though mules are generally a bit smarter than the horses. And by a bit, I mean a lot. Anyway, horses also suffer from, some of the inbreeding. Don't get me started on how Arabians look right now. Anyway, comparatively speaking, kind of like, with dogs and cats, we've bred them into some stupid places. Anyway, so gene flow is between populations that holds a species together genetically. So remember, this is kind of migration and whatnot. And that's a good thing, because new, populations pool up, people move, and we've got literally fresh blood coming into the population. So that's a good thing, going back to the let's keep our bloodlines pure with marrying our cousins and all that jazz that we used to do to all the all the royalty and whatnot, and the ancient Egyptians took it to another level. But long story short, with cheese, keeping pure bloodlines, again, genetically is bad. Gene flow, good. We like it when we can get, again, more genetic diversity is a good thing. So remember, spread your genes around. Well, not your pants. Wear your pants. Anyway, moving on. So again, roadmap to a new species. Usually what happens is sometimes there's going to be somewhere a barrier that splits, and it can be a bunch of different barrier types. We're going to go into the barriers in a minute. But basically, there's a population, there's a gene flow between these populations. But given time, once upon a time, something happens between two populations, one splits off, and there is a barrier to gene flow established. That barrier, whether it be a physical thing, like all of a sudden they went to an island and they can't swim back for whatever reason, or they floated on over there, or there was a volcanic eruption that split a valley into, or something like that. That's a physical barrier. But there's also genetic barriers, and we're going to talk about that because it actually is kind of interesting that, when I say barrier, a lot of people think of physical ones, like, some kind of geologic thing going on, or, immigration to an area that, you can't get back from. Stuff like that. But actually it can be more nuanced than that, and we'll get into that in a minute. So eventually, over time, the gene flow continues between these two groups, but this group changes. Now, this is not exactly, like I said, a swift thing. However, we have seen it over several generations in birds, and we had a husband-wife science team duo actually see this happen on the Galapagos Islands, and they wrote a huge paper about it. And so we've seen this literally happen. So when people go, nothing, you don't see it. No, we've seen it. It's actually happened right before our eyes, several times and several times since. So this is not something that has to happen over millions of years, but it's still going to take several generations to get it done. So, population begins to genetically diverge, and over time, even if that barrier goes away, you can't have any inbreeding. Now, this is basically where, horses and donkeys are. Donkeys have gone one way, horses went the other way, and they can still interbreed, but they don't produce viable offspring. These guys, are basically just not interested in each other. Usually if we put them together, they'll start to compete. And again, competition is not, one-on-one, I beat you, I get all the food, you die. Although it can be that, but there's other, again, nuances. But now that it's reproductively isolated, they can no longer mate, they can no longer do anything, and they're not terribly interested in each other. And you see this a lot in insects, too. Insects are kind of interesting, and again, it's a different type of barrier, where they have a very much lock and key system with their breeding. In other words, literally, their delivery system for the sperm only fits into their female species and cannot fit into other female species whatsoever, even if they're like two sets of different grasshoppers, just as an example. Insects definitely took it to an extreme, made it definitely a lock and key mechanism. So one species of grasshopper really can't mate with another species of grasshopper because their sexual organs are so different. Insects went that weird way. They'd be weird. We'll get into them later. They're kind of fun in a weird way. But anyway, so again, how do we get two new species arise from one foundation species? And there's a ton of different ways. There's not just one way. There is a lot of different ways. But we're going to split it up into two overall categories, which is allopatric speciation, which is called other country. So imagine, geologic separation to form the new species. So physical barriers and whatnot, geologic things. So allo means other, patric means homeland, so allopatric means they moved somewhere or a geological event happened and now the population can't have access to each other whatsoever. Sympatric speciations is the other one I was talking about a little bit earlier, with the fact that basically there's nuances, basically these two species start from the same species but can split from each other even when there's no geologic barrier. It can be timing, it can be a whole bunch of different things. And so sympatric speciation means same country, sim means same, patric means homeland. Hello, this is Patrick. And involves speciation occurring within parent species remaining in one location. So the species are hanging out together, but over time, they start drifting farther and farther apart because of timing or something else, and we're going to get into what that is. So again, an allopatric gene flow is interrupted when a population is divided into geographically isolated subpopulations. So we've seen this. We've actually seen this in lakes. For example, the water in a lake may subside and form two lakes with separated populations. We've actually seen that. We've actually recorded that. Again, island hopping is a major thing, which, again, we've seen it over and over again with the finches on the Galapagos Islands. That's what I was talking about, that married couple. Allopatric speciation can occur without geographic change, and individuals colonize in a remote area. For example, flightless cormorant of the Galapagos likely originated from a flying species from the mainland, but then they sat down and said, you know what, I like it here, so who needs to fly? It's kind of like, I don't blame it. It's like driving in Atlanta. Ugh. Anyway, some places are easier to drive in than other places, as I'm sure you probably know. So, same thing with there. There's just like, oh gosh, I just flew in and boy, my arm's tired. And the cormorant said, maybe we should just give up on this flying thing and live the island life. And they did. So anyway, allopatric speciation. So we've got a few different types. We've got dispersal, when a few members of a species move to a new geographic location. So again, they're just like, oh, there's just too many of us here. We're leaving. Goodbye. And that happens a lot. Actually, some plants bank on it. Try to get their kids as far away from them as possible through using the wind and whatnot. In some place, birds do it all the time, which is why we have so many different types of birds all over the place, because they're always flying off. Vicariance is basically when a natural situation arrives to physically divide the organism. So like I said, vicariance is splitting the range. Like, for instance, it may not even be geographical, like, all of a sudden but there might have been a middle population that was bridging the two populations here, and they went extinct, and now they're completely cut off from each other in that way. Again, another one could be earthquake. All of a sudden there's a gap, and now physically they can't go over the gap. So think of ant colonies. I mean, imagine a flood, and it puts a river through, a valley, and then ant colonies are completely separated on either end of this new, it doesn't always have to be huge. Some can be small, especially for the small creatures out in the universe sitting there going, oh my god, where did all this water come from? Imagine on an ant scale, something crazy like that. Anyway, adaptive radiation. So many adaptogens evolve from a single point of origin, like on an island or an archipelago. So a founder species come out, and then because they really can't get back to where they came from, they just branch out. Like I said, we got these guys that eat insects, these guys that have designed to eat nectar, grubs. They use tools to get their food out. These guys like seeds. These guys like leaves. So again, basically what Darwin saw on the Galapagos and made him go, huh? And ever since, we've been studying all the populations on the Galapagos and going, again, they just decided to split out into biological niches depending on what food they were going after. So they weren't all going after one food source and depleting the island of it, because if they did that, they'd be dead. And that has happened, Some creatures have come in, colonized, and had a failure to regulate their environment, and they go extinct because of that. So, how do they know? Sometimes they don't, okay? Sometimes they really, really don't. And it's true with humans, too. The big, the Moai statues, those big Pacific Island statues, and the big heads that stick up out of the ground. And actually, there's a whole more of a statue below the ground. That entire civilization went extinct because they chopped down all the trees to make these giant moai heads. So again, it isn't just animals. It's like, how did they know? Well, in this case, they kinda didn't. So yeah, it's not, like I said, it's not just animals. Sometimes humans do the same stupid things. And sometimes plants do it, and sometimes fungi do it. No piece of life is exempt from screwing up and going, we just dorked all our resources. Oops. So, anyway, sympatric speciation. So we talked about genetics, and sometimes you get non-disjunction during meiosis. So sympatric, usually the basis of this is, I'm sorry, it's getting darker and darker out. Of course, the storm's coming in. But anyway, so it's basically, remember non-disjunction, where sometimes chromosomes fail to separate? Sympatric usually starts on a genetic level, whereas allopatric usually starts in a physical level in some form or fashion. So sometimes we get creatures that don't have enough chromosomes. It's called aneuploidy aneuploidy, so you don't have the amount. And if you have too many, it's called polyploidy. Now, some creatures, like plants, deal with having too few chromosomes and too many chromosomes way better than we do. We, as humans, don't deal with it very much at all. Polyploidy, we can deal with to an extent, like trisomy 21. That gives us, Down syndrome, but otherwise an individual can live. However, there's other polyploidies like trisomy 13. It's one of the first things you get tested for when you get pregnant. That's why they suck half the blood out of you to do a battery of tests on the baby and the mommy, make sure everything's cool. And one of the tests is trisomy 13, because if the baby has that, they will not survive in the womb whatsoever. They usually die. There's no way around it. There's no way. It's just, if you have trisomy 13, the doctor sits down with the family and has a very sad conversation, because the fetus will die. It's very rare if they get born, and even then they die. So, not cool. So like I said, us mammals, we don't deal with polyploidy very well, but we actually do not deal with aneuploidy at all. If we don't have, when humans have 26 chromosomes, if we don't have our full 26 chromosomes, we just don't fail to turn into a fetus. So if a sperm or an egg has less than the 23 that they should to turn into the 46, did I say 26 earlier? I apologize. Hi Monty. Anyway, and basically what happens is they merge and nothing happens. It's a dud. So a lot of mammals, we don't deal with this. However, other creatures, like I said, plants love, it's how we make giant strawberries, is we just force strawberries to have a ton of chromosomes by forcing them to go through non-disjunction. And then we get a giant strawberry that you dip the chocolate in, and then you go, strawberry chocolate, yummy. So anyway, so that's basically, too many polyploidy, not enough aneuploidy. However, this gives the basis where if you've got one polyploidy creature and another polyploidy creature of the same polyploidy, guess what? They could breed and make a new species. And that's reproductive isolation. It's very, very common in plants. This happens in plants a lot. And we've used it to actually make our own plants. So this is not something that's like, but it's very rare in animals. We don't really do this with animals. This majorly leads to, of course, thunderstorms coming. Monty, did you call this? So anyway, long story short, with cheese, this gives rise to reproductive isolation, especially in the plant kingdom and other kingdoms, not so much in animals. Now, there's autopolyploidy, which is having double the chromosomes, means it can only mate with itself, and then gives rise to a new species. We have seen this in lizards. The reptiles can do some interesting things. We've seen this with lizards, I believe, down in New Mexico. Allopolyploidy is when two species combine within 2 generations to create a fertile offspring, which is a new species. So again, sympatric speciation usually starts on a genetic level and then builds up, especially in the plant kingdom. Now, I said plant kingdom does this. Yes, it do. And we have seen it in reptiles. We have seen it in other places too. And now I have hiccups. Hold on. Hopefully that works. No, anyway. It usually doesn't. Anyway. And that brings us to another way, and this is usually where all us animals start doing this, is reproductive isolation. This is where a divergent characteristics affects a species to interbreed and create a new species. So there's different types of isolation types. So one is the prezygotic barrier, which is basically this. It's basically where the egg and the sperm meet, but they don't really, they merge, but something might happen that makes them different. That's called a prezygotic barrier. There's a postzygotic barrier, which is usually after the egg and the sperm meet and get fertilized and can lead to a new species. Again, things like prezygotic non-disjunction. There's a good example of a prezygotic. So something like non-disjunction, stuff like that. Postzygotic mutations can arise. That's another one right here. So all the creature is forming in mommy or not in mommy or in an egg. Like where Monty was formed, he was formed in an egg. It's a funny thing with ball pythons. As they hatch, they get out of their egg and then they sit in their egg for a while, wondering if they should get out of the egg or if that was a big mistake. So anyway, temporal isolation. So this is timing. Birds are definitely like to do this a lot. Sometimes the birds, there are certain species that like to sing all morning and then they shut up, and that's their time of the day. And then there's birds that like to sing in the afternoon and then they shut up. Again, that's their time of the day, and depending on when their time of the day is to sing and attract mates, that's when they'll mate, but not during, if a bird hears, they're used to morning time, they're all morning people, and the night they don't talk to the night people because they're too busy going to bed and probably going, shut up, we're trying to sleep over here. So temporal isolation, you can see that with bird singing. You can see it with plants. There's some plants that, like, for instance, morning glories. What they like to do, they open up in the morning. That's when you're going to get pollinated. And then we have the other ones that open up in the evening, and they get pollinated by a different group of pollinators and at a different time. So that's when they'll be open to getting pollinated, and that's a timing thing, and that can lead to completely different species just based on timing. So so habitat isolation occasionally, like if a river runs through it, but also layers. There's strata, and we're going to talk more about this when we get into ecology. But like, for instance, if you take a rainforest, it is a biome, but there's also layers within the biome where creatures stick to that place. There's creatures that only live on the ground or only live in the canopy and never come down, or in between. So, there's the ground, there's the brush, there's the undergrowth, and then there's way at the top, there's the canopy. And these vertical levels of a biome can completely and utterly make it so species don't even see each other. So there's, creatures that live in the canopy, they'll never go down to the ground. Not unless they're dead. So they won't see anybody down there to mate with. So that's a habitat isolation. They're all in the same biome. They're all in the tropical biome, but that doesn't necessarily mean they're ever going to meet anybody in another area. So again, behavioral isolation, again, we can use bird singing back to this. It's not only just timing, it's also the songs. Certain species like certain songs, except for mockingbirds that pick up everybody's song and then make remixes it. So they're the ultimate remixers. So I'm going to pause here and turn on the light. That's a little better. Okay, where was I? So, and again, behavioral isolation. So certain birds sing certain songs, and because they sing certain songs, it's not, it's generational. It's somebody's listening to a song and they're like, ew, I don't like that music. I get it, Everybody's got preferences, except in birds that kind of led to speciation. Somebody was singing country and somebody else who's a rock diehard was going, yeah, no. And the metalhead birds in the corner going, yeah. Anyway, so, gametic barriers, again, this is, getting them in. Mechanical isolation, again, I was talking about with the reproductive systems of male and female genitalia are very, very unique. Special damselflies, this is all damselfly genitalia. Yeah, how does that even work? Well, it only works with their species of damselfly, nobody else. So that's a mechanical isolation right there. Hybrid inviability, so you make a hybrid like horses and donkeys, but it doesn't really do anything. So bada boom, you have to get more mules, you got to get horses and donkeys together. That would be hybrid inviolability. Another example of this is the liger, although tigers and lions, if you put them together, they're not really in the mood, okay? Even if you set romantic candles. We have to make ligers in the lab. We have to get the sperm and the eggs and put it together in a lab and then put it in a mommy. It does not work in practice. Like I said, lions are very... socially adept. They're more social. And tigers, not so much. So when you put them together, usually the tiger beats the snot out of the lion because the lion's like, you kitty? We friend? And the tiger's like, no, get out of my life. Anyway, ow. Okay. Watch the internet go out in the middle of this. Anyway, so habitat influence. Again, there's a lot of habitat influence. Things like heat or temperature can really mess with things. My favorite is with fish. We actually, when I was going to Western Carolina University, they were doing some really groundbreaking experiments by heat zapping fish eggs to change the entire batch into male or female. And it led us to understanding how temperature of water really can change sexes. We can literally change the sex of fish when they're in their eggs just by heat or cold shocking the eggs to get them to turn into a boy versus a girl. And that was stuff that was happening at Western when I was going there. Now that I think about it, it was 20 years ago! Ish. Ugh. When did I get old, Monty? Anyway. So that's an example of speciation.
You and me,
Nope. All right, so now let's go ahead and jump into chapter 26. So that was a bit of chapter 24 that I found important. So let's talk about some chapter 26 bits, which is why I have to bring into how is everything related to each other, and how do we try to draw that? Again, keep in mind, it's not as clean as we make it look on a piece of paper. We do this stuff like this to try and keep it straight in our head. In reality, there's a whole bunch of all over the place. So, which is why, of course, I put Pokemon in again. Somebody decided to try to figure out how everybody in the Kanto region is related, which makes me giggle because I don't even, dude, I'm nerdy. And I tried to figure out the biology from the Disney show Gargoyles, but I was really bored that year. This is a step too much for me. But anyway, so how do we do this? So basically, again, Darwin kind of drew this in the beginning. It's called phylogeny, and it's the evolutionary history and relationship of an organism or group of organisms, and we can draw the tree in different ways, and I talked about this a couple chapters ago. You can have the rooted one that literally looks like a tree, or you can have the circular one, which is called the unrooted phylogenetic tree. And Monty's squeezing me. It's okay, dude, we're not outside. Anyway. So, and we do this a lot. This is basically, again, we try to figure it out so we can see it. We, as humans, do better with seeing things in a lot of cases. So drawing it out so we have an actual kind of model to look at helps us a little bit, especially about talking about these things. But like I said, if you're adding in time to this wouldn't be as clean. And even then, we're going to have some gaps. So, we're all related, but some not as much as everybody else. Now, keep in mind, there's two things going on here. We got phylogeny, which is the evolutionary history of a species or a group of related species. And you can see, we're all vaguely related. Not, I'm not related to a fish directly. Really off. But our genetics is all there. We all have the genetics, especially in the animal kingdom. If you look at this bunch right here, even though we all have bones, and even then, it kind of goes back and forth because some of them have no limbs and some of us kept our limbs and all that fun jazz. Fun fact about humans, we actually don't have much of a family anymore. Homo sapiens are actually the only of the hominids that are existing today. I wouldn't say we are the wieners, because I don't think it's like that. I'm just saying that we have no family. We're orphans. Anyway, because the other hominids did not make it to this point. We're the only hominid left. Again, I don't know. We're still trying to figure out how we became the only hominid left. And it could have been evolutionary. It could have been we outcompeted them. Or it could be environmental. It could be all of those things put together. And although it is funny, because we did find that a lot of their genetics, now that we've found, specimens and ran their DNA when we could, we found a lot of their genes are inside of our genes. Which means there was some way hey there. Silly us. So maybe we absorbed them. With our powers combined, we became Homo sapien. I don't know. We're still working on that one. Anyway, systematics is a discipline focused on classifying organisms and determining their evolutionary relationships. So, looking at, even though humans and chimps are pretty much 2% away from each other, we're 98% have the same genetics, but they're not remotely related to us. If we had another hominid group running around, they'd be way more related to us than the chimpanzees. And if you've ever seen chimpanzees, never mess with an angry chimp. They will destroy you. They will discombobulate you. They don't outright kill you. They do horrifying things to you because they're pretty intense. Jane Goodall learned they were not happy, slappy, lovey-doveys. They're actually murder psychos that also can use tools. So, yeah. Their whole family is fascinating, because you get the chimps that went one way, which was the ultra-violence, and the bonobos going the other way, which is ultra-love. The bonobos are interesting because they, instead of having fights, they actually hug it out, but with sex, which is really interesting, whereas the chimps just beat the poo out of each other, and by the poo I mean they bite all their fingers off, they rip their eyeballs out. I mean, don't fight with a chimp. If you ever want to look up something to really twist your stomach, look up chimp attacks, because anyway, yep, power just flipped. That was fun. Really? Do you have to do this while I'm recording, Nature? Of course it does. Of course it does. You know. I know. You know. Anyway, now, so speaking in a rooted tree where the branches indicate evolutionary relationships, the point where a split occurs, there's a branch point. And these branch points... I'm going to shut down until the storm's over.
2,000 years later.
Okay, where were we? So, we were talking about trees, and I don't mean the ones that go through photosynthesis. So, we were talking about in a rooted tree, and we were talking about where a split occurs is called a branch point. So, this represents where a single lineage evolved into a new one. So, keep in mind, these don't happen overnight, all right? But, again, we're trying to draw this out so we can visualize it. This isn't necessarily representative of time all the time. So, this is just showing where, we've got evidence that a branch point occurred. So, therefore, this one species hit a point where speciation occurred, and one species kept going while a new one popped off. Now, that lineage that evolved early from the root and remains unbranched, like right here, that's called a basal taxon. And some of these go on and turn into a whole new tree, and some of these don't. I mean, there is a lot, a lot, and we're going to get into a little later down the line. Just how much, things have branched off and, oh, that didn't work. And trust me, it happened a lot. Plus, we also had about 5 massive, die-offs on this planet alone, where the entire population, everything that was living, was almost reduced to 0. So the Earth has tried to shuck us off, and I don't mean humanity, I mean life, about 5 times. And like I said, 2 of those times we can kind of sit there and stare at the plant kingdom going, really, was that a great idea? And the plant kingdom going, Anyway, we'll talk more about that in a bit. But anyway, so long story. So sometimes if you get a branch and then you get more than one branch point off, this is called polytomy, and this illustrates where scientists have definitely not determined all of the relationships. Like I said, we have a lot of evidence, and I do mean a lot of evidence. This isn't stuff where we're just, going, and we've seen it. We've also, like I said, we've got a lot of evidence because we've seen this happen. Now, keep in mind, just because, you see a polytomy does not necessarily mean they all share an ancestor. It does not, they might share an ancestor, but it does not mean they evolved from each other. We're still trying to figure this one out. And we've got a lot of places that happens. Like, there's a lot of places where fossil evidence just wasn't saved and things were lost. And anybody who's worked with computers for any length of time knows exactly how that is. If back in the old days where we had to use the save, we didn't have autosave on everything. We had to hit the save icon 15 times just to make sure. And sometimes you'd do it twice because you'd be like, That was one of my old questions 20 years ago when I was a computer lab specialist sitting there. That was my job when I was in college. To make some money on the side was I'd sit in the Western Carolina University's computer labs and help people. So anyway, my favorite question usually was, did you save? And their usual question was, My other favorite was somebody lost their entire term paper because they had saved, and then they took their floppy disk, because back then we were just getting to zip drives, but we still had the 3.5 floppy disk, which wasn't very floppy, it was very hard. And he went back up to his dorm room and played some music, but he had thrown the floppy disk on top of his subwoofer. And if you know what I'm talking about, the subwoofer is a magnet, so it went... And he came back down to the thing, and he goes, the term paper's gone! And I'm just like, okay. So I'm like, let's retrace our steps. What computer were you on? Check the computer, checked his floppy disk, it was clean. It had been wiped by the subwoofer. And then he mentioned, yeah, I threw it down on top of my subwoofer. And I was like, oh. So, yeah. Remember, big magnets and computer memory storage devices do not mix. And he didn't understand that and yelled at me for a little bit longer. And I was just like, dude, you're the one that threw it down on a subwoofer. You erased your own stuff. Well, I didn't know that. Well, okay, I can't do anything. Anyway. So, it's the same thing. We've lost, just as you might have lost data to corruption at some point, and that's just the way it is, and then you're like, no, I have to do all over again. So has the universe. Especially, so, like, for instance, the lineage of bats. We do a lot through DNA and whatnot, but if we could find a bat fossil, that'd be great, but unfortunately, their bones are so tiny, they don't petrify. They don't turn into fossils. So any creature with teeny tiny bones that didn't get itself conveniently stuck in amber or mud, in a certain way, we're sitting there going, yeah. And that's a lot of creatures. We know we have tons of fossils, but there's also a lot of creatures that were so tiny that they just didn't leave anything behind because their bodies and whatnot were not conducive to turning into a fossil. So we do have, thank goodness for DNA evidence, but sometimes it's nice to have the fossil to go along with it so we can sit there and go, what did this creature look like with bats. So anyway, so just to know, and you've got two lines right here, they're also known as sister taxa, and you see a lot of sister taxas. But basically, those are the points that I'm going to point out here on your family tree or your not family tree. Now, again, phylogenetic trees show patterns of descent, not phenotypic similarity. And we used to get this wrong, and this is still something that, a lot of people get on. Again, when I mentioned what's the difference between, with like, for instance, all mammal bones, we pretty much have the same bones. They just come in different shapes and sizes depending on what's going on. Versus, why is a shark and a dolphin shaped similarly? And it's because of physics. Because that's the shape that works the best for what they do in the ocean, which is both of them being top predators. So, long story short, we, a long time ago, used to sit there and look at things and be like, oh, well, this dolphin and this shark are shaped similarly, so therefore, party on. And no. No. So, again, phylogenetic trees do not indicate which species evolved or how much change occurred in a lineage. Again, these are to show us or give us at least an idea in our head how everything is related. It is not necessarily telling us where, when, and how. Just giving us a start point so we can kind of conceptualize it. So keep that in mind. Keep in mind we can also rotate these. So this right here is just as correct as this one right here, even though we've rotated it all over the place. So just keep that. We prefer this one because it looks better. But you will see some that are kind of rotated, and that there's reasons for that. So don't get it, kerfuffled if you see this and go, yeah, that makes sense, but then see this and go, ugh? We can rotate the branch points. And sometimes we do that, A, to save paper, and B, to kind of go off on clades, which you're going to see in a minute. We're going to talk about clades. So also, it should not be assumed that a taxon evolved from the taxon next to it. So just FYI, chimps and humans may have had a distant ancestor that was neither a chimp nor a human. Keep that in mind, because humans did not come from chimps, and that's why I really get annoyed when somebody sits there and goes, well, we didn't come from no monkey. Yeah, I know, we didn't come from monkeys. No, evolution has never said we came from monkeys. at all, because monkeys are their own way out their sister taxa to our human taxa. So just FYI, again, yeah, you can agree with them. Yeah, we didn't come from monkeys. Monkeys are their own separate thing, their own separate track, way, way, way back. At some point, we might have had a distant ancestor that split at one point. One species went one way and another species went another way. But I assure you, we are nowhere, we're somewhat related, but not that related to monkeys. And trust me, as somebody who's worked with monkeys, be thankful for that, because they're jerks. Anyway, everybody's like, but monkeys are cute. Yeah, until they bite your fingers off. I've got stories. Again, if you want some stories, I'm going to be hopefully sending out a thing soon, so that way you can tell me what times work for you and I can have some office hours with you. But anyway, so don't be fooled. Remember the difference between homology and analogy. Again, analogy is just they have a similar shape. Usually that's for physics reasons. Again, why I bring up why fish and sharks and dolphins, even though they're not related to each other, they all have similar shapes, because that's what works the best in the water to get around. Homology, on the other hand, again, if we go from the wolf ancestor and then we get all these dogs, again, we can see the wolf ancestry in some species more than others. Although I have to say the Chihuahua got the anger gene, and what is it? My brain just died. And the husky got the looks. But anyway, so yeah, we they can all track back to a wolf ancestor, because if you look at a dog skeleton, you look at a wolf skeleton, you can literally sit there and say, yeah, these two are definitely related. So anyway, and I'll get into some more fun facts where dolphins came from. So yeah, dolphins were, and I use this very loosely. There was a wolf ancestor, not the wolf ancestor, but there was a wolf-type ancestor that did get pushed back into the ocean off land, and over time turned into what we now know as dolphins. So dolphins, the reason they're predators, the reason they're actually have a way, way, way back, very loose relation to wolves. So yeah, next time, look at your dog, look at a dolphin, and then go, wow, that's weird. Anyway, we do have the skull evidence for that, too. We have literally where there was a wolf-type ancestor. Again, it's not a wolf, but a wolf-type ancestor, that we can see the change in the skulls over time from the land mammal into the ocean mammal, which is really fun. And it definitely shows why dolphins are very dolphin-y. Anyway, so phenotypic and genotypic similarities due to shared ancestries are called homologies. Again, this is important. Organisms with similar morphology or DNA sequence are more likely to be closely related than those that are vastly different. For instance, dolphins and sharks. Again, they have a similar body type, but that doesn't necessarily mean they just, have similarity to what's called convergent evolution, where basically physics says this shape works the best in this environment, and basically life forms go, yeah, that makes sense. And therefore we get dolphins and, sharks, even though they're not very, technically everything on this planet is related, but not that much. So in this case, they're definitely not that much related. So anyway, when constructing a phylogeny system, you need to distinguish whether it's a result of a homology or an analogy. And that is the thing. That is the thing that a lot of people get confused. So that's why I please understand that, yeah, this Australian mole, when we use moles in quotation marks because it's actually not in the mole family, and this African golden mole, which is in the mole family, these two have a similar body shape because that's the best shape for digging through the earth. Again, powerful front claws, blunted face. Again, that's the best shape. Physics. So, yeah, here comes physics. So, again, that's why. But this confused us for ages, which is why we got some really interesting dinosaur. I'll show you one in a minute. But, yeah, that's how we got some very interesting dinosaur fossils at some points where people were just putting bones together going, I guess this works. which, nah. Now, in this day and age, we don't just use trees. We have gone over to something called cladistics, which is basically organisms that are grouped primarily by common ancestry. So a clade is a group of species that includes the ancestral species and all of its descendants. So say you're looking at something like spiders. So we split these up into clades, and we got clades nested in larger clades. So think of the Russian nesting dolls, and that's basically what we do with clades. So we have lots of clades. This is where all the fun arguments are. So when they sit there and they talk about clades in some of the videos down the line, this is what we're talking about. So basically it's the ancestral species and all of its descendants. And we're always putting clades inside other clades and outside of other clades, and sometimes rearranging the clades. So if you hear an evolutionary biologist talk about clades, this is what they're talking about. They're taking one big group of related and then trying to put it into another group to see where it fits. And that's cladistics. That's what we're using at the modern time currently in this year of 2025. Now, I might have to redo these videos in the future when they come up with something else. So just FYI, clades are what's going to be modern. That changed from the time I left college and since became a teacher, so it's been interesting. Now we got monophyletic groups, which are containing the ancestor and all of its descendants are clades. Now we've also got paraphyletic groups, which consist of the anthrospecious and some, but not all of the descendants. And then we got polyphyletic groups, which includes distantly related species, but not their most recent common ancestor. A good example of this is currently, I know there was last time I went and I took a course somewhere to freshen up on some things, because I always try to stay up to date. When I was taking a spiders course, found out that there is one guy that wants to stick and I know this seems crazy, horseshoe crabs in the spider clade. And everybody was losing their mind over that because horseshoe crabs are related to spiders. They're in arthropoda. They do have 8 legs, just like, the rest of the arthropods, but they're not quite there. There's the clades currently are, if I recall correctly, are horseshoe crabs, scorpions, and scorpions are actually pretty old. We've bounced back and forth between scorpions over the evolutionary timeline quite a few times. Nature came up with scorpions and went, Scorpion! Scorpion! All right, we'll get into that later. And then spiders. And there are actually an in-between of scorpions and spiders, but they're actually spiders. They're called pseudo-scorpions, and they're in the spider family. So just when you thought you couldn't be any more confused, classification people will make you confused. So those are some of the groups inside of clades, and like I said, this is the most modern understanding we've got right now. We took the trees, we've turned them into clades, and now we're shifting the clades around one huge group at a time with the ancestor and all its descendants. And that's how we can kind of plug and play, hopefully until we figure it out. But I sincerely doubt that's ever happening because A, people love to argue constantly, and B, we're always getting new evidence for something, and then it hits, and then we're all like, what? And then we have to rearrange the clades again. So this is normal. We like the plug and play aspect because remember, if science isn't changing, we're not doing science. But remember, science needs to change with proper evidence, not just because we said so. So to sum up, phonetics is a method of classifying organisms based on structure and morphological characteristics that are observable. Cladistics is basically the method of classifying organisms based on their ancestry and evolutionary relationships. And that's why we go with cladistics now. We used to do this one, but unfortunately, if you're looking at just observable characteristics, you get yourself into some piles of trouble. So keep in mind, this is what we started with. This is what a phenogram is, but cladistics is where we're at now. So we started here and went to cladistics, which is cladograms and phylogenetic trees, and we have a lot higher accuracy with this and a lot more remember, if you can reproduce it, you're doing science. If you can't, somebody was lying to you. Anyway, and so that's why cladistics. We started with this because that's all we had. All we had was observable characteristics for the longest time until we figured out DNA and then went, oh, we can go back and DNA test everything. And that's when we found out that even in the cat family, we've got some really interesting, like cheetahs. You think, oh, aren't they related to other cats in Africa? No, they're actually more genetically closer to our mountain lions. Weird. So again, that's why we had to go over to cladistics, because it's based on actual evidence, not just us looking at it and going, dolphin and shark are same shape, so they must be related. No. So again, if you want more how to draw them and how to do them, watch my favorite Mr. Anderson right here with Bozeman Science, or my favorite knockoff Brad Pitt. He does a really good job explaining this. So, now, one last thing I want to mention for this chapter, and that is the molecular clock. So, we can actually use mutations and figure out when they happened on the timeline of Earth. This is actually really cool, but it also helps us estimate absolute time of evolutionary change. Because if you can figure out where the mutation is, you can figure out when that happened. And that is called the molecular clock. It's basically an approach used to estimate the absolute time of evolutionary change. So, the estimate is based on the observation that some genes appear to evolve at a constant rate. Now, evolution happens in fits and like, you know, me trying to drive a stick shift. It's sad to watch. It's like, you know, sometimes it shoots forward when there's plenty of resources and the Earth has plenty of time, and then sometimes it stagnates, and sometimes it shoots, and sometimes it's like watching me drive a stick shift. Bad. So anyway, however, we can estimate on, you know, that some genes do show up at a constant rate because some mutations are always happening. And our body is always going back and fixing them. So if we know that, we can then go back, and there's a bunch of math involved in this. Please don't expect me to explain it. All the math, because sadness. Anyway, the molecular clock is calibrated by graphing the number of genetic differences in a gene against dates of branch points known from fossil record. And it really helps if it wasn't just the fossil record, but if we got some DNA there too. Because remember, we're carrying around a lot of DNA we don't use. It's not junk DNA. We can use it. It can be turned on and off, but we're actually carrying all of the DNA that our ancestors gave us. So if we look at our DNA and then compare that to an ancestral DNA, and then you can see where the mutations are, you can be like, oh, and plot out the timing of when the mutation happened. So this inferred rate of change is used to estimate the dates of the fossil record that's not even discernible from the fossil record. So keep that in mind. We don't need to just sit there and somebody's always like, well, carbon dating is bad. Yeah, carbon dating's not great. It gives us a ballpark, but we have other types of dating. And this is the most modern one, is the molecular clock. This gives us dating based on the rate of change in mutations of a genome, which is a lot more reliable than currently, you know, sometimes looking at how the half-lives of certain elements happen in. So that gave us a leg up to this. So this is the molecular clock. Yes, you'll have one question on the test about this. Wink, wink, nudge, nudge. All right, so anyway, so that's the molecular clock. And be aware that's basically what we use right now to try and figure out timing of where these popped out. Again, it's kind of interesting to see all this. And, you know, when you take a modern lineage and another modern lineage, and you can see that there's two differences between them, and then you figure out when that happened, which I think is amazing. Whoever figured this out, stroke of genius, which is probably several people, but at the same time, rock on. Rock on. So again, phylogenetics, Mr. Anderson here goes over it. Good video. Highly recommend you watch, especially if you want some more on how everybody's kind of related and how we use the trees and everything to kind of visualize this stuff and go over the terms again. So highly recommend. Oops, didn't want to play that. And with that, we are done and dusted with chapter 26. Sorry for the interruption there. And I will see you next time when we hit up. Now we're going to go ahead and move into going through all the domains and then the kingdoms generally, going over some major points in evolutionary history for each creature-ish. Not, if we did each creature, we'd be here forever. But anyway, but we're going to start with the one thing that maybe you covered in 111, maybe you didn't. I don't cover it in 111, but I like covering it here, and that is the weirdo group of viruses. I like covering viruses. I think they're weird because they're not really in anything we're talking about. They're that weirdo zone of the biology dimension, and I'm going to talk about them before we jump up to archaea and bacteria, because I think they need some love, but not in that way. I mean love as in I distantly think they're neat, but I don't really want to meet them much because every time I get sick, I'm not a fan, and I'm sure you're not either. So anyway, next time we're going to hit chapter 19. So yeah, we're doubling back in the book. We're hitting chapter 19. We're going to hit viruses real quick, and then we're going to jump into archaea and bacteria because it's fun like that. All right, with that said, I'll see you next time. Bye!
But wait, there's more!
One more thing. I completely forgot because I wasn't going past that one video, and I thought it was the end of the notes because usually I end my notes with videos. So, my bad. Welcome back. Real quick, I want to talk about something that is absolutely huge and invaluable if you're going to go forward in biology in any form or fashion, and that is a dichotomous key. I need to talk about dichotomous keys. If we were doing a face-to-face class, I would be having you use dichotomous keys pretty much for the rest of the semester in lab. Reason being is because dichotomous keys are extremely important, and probably a little later when we talk about the plant kingdom, I'll have you go outside and find different bits and use a dichotomous key to figure out what you just grabbed. So, what is a dichotomous key? It is basically our way in biology used to identify organisms. Di means two or twice, and economist basically means choices. So it's kind of like, and I don't know, this is dating myself, but there used to be these books called Choose Your Own Adventure books. They've come back. If you're into Five Nights at Freddy's, again, I'm being my nerdy self here, but Five Nights at Freddy's had just put out some horror books that's a choose your own adventure. And basically what it is you'd go along and you'd get to this cliffhanger in the book as you read, and you could choose, what path the character takes. It's like, does he go open the door and walk through the door? Or does he, he was originally used in fantasy books. I used to hate them. I read several and went, this is horrible. Because I always made the worst choices, apparently. But it would always sit there and say, go to page 274 or go to page 492. I'm just using random numbers. And you do that choice, and usually either you'd succeed in choosing the correct path in the book and get the winning ending, or your character would die in unreasonable ways. So that was a choose your own adventure book. This is like the biology version of that, in the fact that it's basically you're using it by looking at whatever specimen you have, and you have two choices, and you've got to make one of those two choices and then go wherever the two choices tell you until you get to hopefully the right species that you're looking at. And I have used many dichotomous keys, many. And the ones I have used, even the simplified ones sometimes will drive you bonkers because the description and or images are of perfect organisms, not of always the ones you catch. So sometimes it's tearing your hair out in the middle of a lab trying to identify what creature you've got. And if I was back in my room, my office, I would show you my spider dichotomous key, which is about that thick, and that's just for North America. Yeah, it's a big thing. And the problem with that one is, if you catch a juvenile spider, you can only get down to probably the family. You probably can't get into exact species because they need to be their adult form. And a lot of times spiders don't hit their ultimate morph until they're usually a juvenile type morph until they hit their ultimate morph, and you're not always catching spiders in their final form, and that's a problem with usually doing insects in some form or fashion. Either it's keyed to the adult form and not the juvenile form, because the sexual parts aren't usually ready yet to figure out what it is. As I mentioned earlier, insects and arachnids both have very, 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 very specialized sex organs. So sometimes you have to wait until they're mature to figure out what species it is, because two species can look very, very similar otherwise until you flip them over and look at their lady bits. The guys, not so much. Guys are pretty easy on spiders. They usually, if they have two big bulbs at the front, that means it's a boy. But anyway, on their pedipalps. So anyway, long story short, I've used many dichotomous keys, and if you're going to continue on in biology, you too will get bonked in the head with using a dichotomous key and tearing your hair out and trying to figure out how to work it. Now, on the whole, a dichotomous key is pretty easy to use, but it's when, like I said, you get a picture and you're like, or description, and then you look at it and it could be either or, and that also, I've also had to do aquatic insects, and we do them as juveniles. It's easier than their adult form, unlike in spiders. And sometimes after you catch them, they have missing bits, and because there's some parts like their gills that stick out of the body, and they fall off real easy. And then you're sitting there going, well, does it have 4 gills or 6 gills and 2 fell off? It's rough. But anyway, this is how you use a dichotomous key. And I did add in, that you should be doing some work on one of the dichotomous key videos from the Amoeba Sisters. But basically what's happening here is that you have 4 birds here, okay? And you can see they each have different beaks. They're the same species, but same family, but different species. So we've got to figure out who's who. Like I said, this is a bare bones basic just to get you used to how it works. Doesn't matter what bird you start with. So let's go ahead and start with bird W. No matter what you start with, you always go back to number 1. Doesn't matter. Doesn't matter until you know your dichotomous key, like the back of your hand. Like, I never went back to number 1 with my spider. There was another further on down that I would always go back to. But that's if you know your dichotomous key really well and you've been using the absolute cheese out of it and you've got, like, the first bunch memorized, which happens. But rule of thumb always states, no matter what specimen you work with, if you've got like 2 twigs and you start with 1 twig, you always start at number 1. If you pick up the second twig and start working on that, you always go back to number 1 on the dichotomous key. No questions asked, do not skip, do not pass go, do not collect $200, as the Monopoly says. So, right here, you look at the beak, it says you always have 2 choices. So it keeps it to 2 choices. It makes it simpler so you're not sitting there looking at 15 choices going, well, I don't know. It keeps it simple between 2 choices. That's why the di and dichotomous key means 2. So you got 1A and 1B. So the beak is relatively long and slender. If you look at that, that is not a long and slender beak. That's a honking beak. And then the beak is relatively stout and heavy. So yeah, that's pretty stout, I'd say, and heavy. Another problem with dichotomous keys is sometimes they will use terms that you may not be used to in describing things. And either this is when you turn to somebody who's used the dichotomous key longer than you, or you Google it. Thank goodness we're Googling. I didn't have it back in my day. So I'd usually have to go harass somebody. But stout and heavy means it's big honkin'. So it says go to 2. So you go to number 2. And then you go, again, you look at just these 2 things. The bottom surface of the lower beak is flat and straight. Yeah, that's pretty flat and straight. But before you sit there and go, oh, that's it, always read both choices. That's the number 2 rule. Number 1, always start at number 1, unless you know the dichotomous key really well. Number 2, Always read both choices even though you think you found the answer, just in case. And this usually is where sometimes you start second guessing yourself. But again, usually you're working with somebody that can come up and help you, which is really nice. The bottom surface of the lower beak is curved, go to 3. So, as you can see, this is straight. So, we could probably say for this purpose, since there's only 4 birds and we only have 4 answers, this would be Geospiza. Geospiza. So, he's a Geospiza. So, again, if we do bird X, again, we would start back at number 1, even though we could say, hey, that's a big honking beak. Still, we go back. Number 1, the beak is relatively long and slender? No. The beak is relatively stout and heavy? Definitely. Go to 2. The bottom surface of the lower beak is flat and straight? No. It's curved. And the bottom surface is curved. Go to 3. So, the lower edge of the upper beak has a distinct bend. Now that looks pretty straight. The lower edge of the upper beak is mostly flat. There you go. Platyspiza. So again, even though sometimes you might be like, I already know, I'll skip. Sometimes that's good for like if you're, like I said, always please start at number 1 until you get to know your dichotomous key really well. And you can identify these things off the bat. But usually, as a novice, you always go back to number 1. You always read both choices. And then whichever is the most, and sometimes you're going to have to back up. And this is one thing, it doesn't show in this because this is a really simple version and very easy because there's 4 birds and clearly there's 4 answers. But like, there's going to be one I might give you later to do twigs where you're going to go outside and snip a couple of twigs off of a tree, a deciduous tree, not a pine tree, not a bush, and come back inside and sit down and try to figure out. Now, you may already know because you've looked at the leaves and you know trees really well, especially in your area, but I'd like you to use the dichotomous key. And half the time when you're looking at it, I'm going to tell you right now, sometimes you're going to be looking at it and either you're going to doubt yourself or you read both and you're like, it could be either. Sometimes when you sit there and you read, it could be either, that's when you flip and go both directions. So you choose one direction, and if that goes nowhere, you go all the way back to whatever confused you, like say it was number 3, and you go all the way, and you go, choose A, because it seems likely, and you go all the way down, and nothing seems to match, you go back to 3. This is where you make little notes all over your dichotomous keys, and then you go B, and then follow it, and then half the time, sometimes, hopefully, you'll be like, oh yeah, there's there. And that's the thing, it's just, you've got to be willing to go back to, previous numbers if you get lost, previous numbers if you're, if nothing's working out. Again, using a dichotomous key is not great, but it's the best thing we've got. Even AI really can't help. Sometimes it helps to narrow things down, but AI gets it really wrong on occasion right now. And like I said, I'm happy for a technology. It'd be nice to take a picture and it instantly tell me with 100% accuracy. But even with the technology growing exponentially as it is, that's never really going to happen. It's nice to use. Sometimes it sets you on the right path, especially when you're using a dichotomous key and you're getting a bit lost and you don't have an expert sitting next to you going, hey, do this. So I always use a combination. And remember, when it comes to using AI, it's good to use, but always verify. Go somewhere else, a dichotomous key, or go to somebody who actually knows and say, oh yeah, this is XYZ. So, dichotomous keys, they are indispensable. They are a thing that needs to be used in biology. Even with AI advancing and whatnot, I don't think we'll ever get away from them. They're the best way to figure out what's what when you've got specimens. And you're going to be using them a lot if you continue on down a biology track. Not so much in the labs. If you're just sticking to microbiology and stuff, you may never see a dichotomous key again. And that's fine. But if you want to go into field work again, dichotomous keys, you're going to own like a bunch by the time you're done. I have several. My most hated one is my aquatic insects one. Anyway, I like the spider one though. But anyway, like I said, it's very verbose, but it always gives you two things, and sometimes, like I said, by the time you're done, you're tearing your hair out. I'm going to have you use a couple of dichotomous keys going forward. I have one Labster lab I set up for you for that, but I also have other labs where I'm going to ask you to go outside, find something, and kind of identify using dichotomous keys to get you used to how to use a dichotomous key. With that said, that's a dichotomous key. Again, here's the Amoeba Sisters. I think I've got that as an H5P for you. Nope. And then, I believe Taxonomy.
Taxonomy.
Yep. And now, so, you might be going, hey, didn't we skip over chapter 25? We did. There's a video, and I'm going to show it. Well, I can't show it to you, or else YouTube strikes it for me copying an entire another video. It's one of my favorite videos, and I'll actually talk about this. It does sum up chapter 25 in the first 5 minutes of the video. And it's one of my favorites. However, it does have a lot of swear words in it, so if you're not, into swear words, but at the same time, it's a really good video. Somebody did make a PC version, but it's got a lot of humor. And it's one of my favorite videos, and it's the history of the entire world, I guess. Highly recommend. It's really long. We only care about the first 6 minutes. So if you watch about the first 6 or 7 minutes, that's pretty much all of chapter 25 that you need to know. I don't even have questions on it. But long story short, yeah, that's the first 6 to 7 minutes is chapter 25. Also, some other important points of what we're going to be going down the road. And I'll talk about it again in the next lecture, a little later when we talk about dun 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 bacteria. So, with that said, now I'm actually done with this lecture. So, thank you, and have a good day, and I'll see you in the next one.